Good day. Today we're going to talk about antifungal drugs. Antifungal drugs act against fungi. Let's recall the characteristics of fungi. Fungi is a eukaryotic organism that has true nucleus. They do not contain chlorophyll and they have cell walls. They also produce filamentous structures and they are able to produce spores. They also contain ergosterol. When fungi passes the resistance barriers of human or animal body, it can establish infection, and that is called mycosis. To know more about fungi cell, this illustration shows us the cell membrane and the cell wall, and also the antifungal drugs that acts on them. Fungi have rigid cell walls composed of a polymer of N-acetylglucosamine rather than peptidoglycan of the bacteria. The fungal cell membrane contains ergosterol rather than cholesterol found in mammalian cell. These chemical structures are useful in targeting chemotherapeutic agents against fungal infections. Based on the illustration, we have here azoles, drugs that inhibits ergosterol synthesis pathway that we will discuss in a later. We have also flucytosine that inhibits the DNA synthesis of fungi. And we have here polyins that disrupt membrane structure and function. Moving forward, this illustration shows specifically the targets for antifungal agents. We have here polyins. An example is Amphotericin B. It binds to ergosterol and form pores or channels. We have griseofulvin that acts on the microtubule system of the fungi, and flucytosine that acts on the DNA. We have also, on the other hand, the azoles and the allyl amines. They act on the lanosterol and the squalene pathway in order to halt or to disrupt the production of ergosterol of fungi and weaken the cell membrane. We have, on the other hand, glucan synthase inhibitors such as echinocandins. The emergence of fungal pathogens makes the development of antifungal drugs a medical imperative in the recent years. It also motivates scientists to conduct discovery on numerous investigation to understand not only the structural families of this drug but also their biological targets that can provide rational means for evaluating the merits and the selectivity of new agents for fungal pathogens and differentiate it from normal cells, which has resemblance. An equally important aspect of modern antifungal drug development takes a balanced look at the problem of drug potency and drug resistance. The future development of new antifungal agents will rest with those who employ synthetic and semi-synthetic methodology, as well as natural product isolation, to tackle these problems with those who possess a clear understanding of fungal cell architecture and drug resistance mechanism. Hence, developments in structure-activity relationship or in medicinal chemistry spark curiosity of investigators who might discover the following new derivatives, new scaffolds, repositioning of other drugs, repositioning of antifungals, or discovery of synergistic combinations, or discovery of natural products and biosynthetically made derivatives. In our lecture, we're going to have structure-activity relationship discussion of these different antifungals. We will begin with polyins, followed by azoles, allylamines, and glycofulvins. These are the different antifungals that has established structure-activity relationship. To begin with, we have polyins. Polyins act directly on the fungal cell membrane. It creates a pore that will allow the Amphotericin B, an example of polyin, to enter the fungal cell. This is illustrated with the color red. Here, this one represents Amphotericin B creating pores on the fungal cell membrane. The structure of polyin has microcyclic lactones with distinct hydrophilic and lipophilic regions. These are produced by Streptomyces species. It has hydrophilic side chain that contains alcohol, carboxylic acids, and sugars. It has lipophilic structures contain the pharmacophore, which has 4 to 7 conjugated double bonds. The number of double bonds correlate directly to activity and inversely to toxicity. Therefore, the higher number of double bonds, the higher the antifungal activity and the lower the toxicity. An example is Amphotericin B, which is 10 times more active and which can be taken systemically. So for our polyin antifungal, it has affinity to ergosterol containing membranes. Then it inserts its structure into the cell membrane, disrupts its function, and leaks cell components by forming a pore. In this figure, we have here the hydrophilic structure and the pharmacophore, which is the conjugated double bonds, which is the lipophilic part. The Amphotericin B, it has 7 conjugated double bond, while the other polyins such as Nistatine has 6 conjugated double bond. So Amphotericin B is more active and more toxic. Most polyin antifungal drugs, they contain macrocyclic lactones that is varying from 12 to 37 atoms in size. So for our Amphotericin B, it uses its structure by interacting with the hydrophobic part of the fungal cell directly on the ergosterol. It will form a pore. Once it forms a pore, the potassium and other small molecules will leak out or will be lost through the pore, causing cell death. Several Amphotericin B molecules binds ergosterol in the plasma membrane of sensitive fungal cells. The pores that are formed are called channels. It requires hydrophobic interaction between the lipophilic segment of the polyin antibiotic and the sterol. The pores disrupt membrane function, allowing electrolytes and molecules to leak that eventually will cause fungal cell death. For Amphotericin B, it is used for patients with progressive and potentially life-threatening fungal infection because of its potency. An example of this infection are aspergillosis, streptococcosis, blastomycosis, systemic candidiasis, and coccidioidomycosis. Amphotericin B is considered as the broadest spectrum of all antifungals. Amphotericin B is also known for its potent nephrotoxicity activity. Another example of polyin antifungal is the Nistatine. Nistatine has almost the same structure with amphotericin B but differs in the pharmacophore in the lipophilic region of conjugated bonds. Nistatine only have four conjugated double bonds while amphotericin B has seven. Remember that number of conjugated bonds is directly proportional to the antifungal activity and indirectly proportional to the degree of toxicity to mammalian cells. Hence, amphotericin B has the highest activity and the lowest toxicity, and Nistatine is just intermediate. Nistatine is still considered as antimycotic polyin antibiotic obtained specifically from Streptomyces norsei. Nistatine is a polyin antifungal to which is applicable for molds and yeast infection, such as Candida species. Nistatine has some toxicity associated with it when given intravenously, but it is not absorbed across intact skin or mucous membrane, and it is considered relatively safe for treating oral or gastrointestinal fungal infections. It is only insoluble in water and sparingly soluble in organic solvent. It can be administered orally for oral thrush, intestinal and monilial infection, and it is the drug of choice for vaginal and cutaneous candidiasis. It can be administered in vaginal tablets or vaginal suppository. Sometimes it can be combined with iodochlorohydroxyquine.